So, we will begin. Like I told you, it is very, you approach this topic with an overall idea that DNA repair mechanisms basically aim to restore the structural damage inflicted by either radiation or chemical agents or by some spontaneous mutations or whatever. So, that is the aim. How do they do it? Basically, you can divide that into single strand breaks and double strand breaks. Single strand breaks are relatively easier to repair because you have another strand which will obey the Watson and Crick base pair rule. So, you have a substitute. So, you can repair it more or less completely to restore the structure very clearly. So, that is the idea. Double strand breaks are slightly difficult. So, we saw the mechanisms, three mechanisms, the base excision repair, nucleotide exchange, excision repair and so on, which deal with that. Now, we will pass on to how the double strand breaks are taken care of. So, one of the most important mechanisms, pathways, you can say, is this homologous recombination repair. All of you know what are homologous chromosomes. I already told you that we receive one allele from father and one allele from the mother. So, we always have that. So, double strand breaks can be, of course, can be caused by reactive oxygen species or radiation, bleomycin, anthracyclines and some topoisomerase inhibitors. So, these are some examples. Even during normal progression of the cell cycle can also have some errors, and that can also cause double strand breaks sometimes. Obviously, failure to repair this will result in mutation, gross chromosomal changes, aberrations, genomic instability and eventually cell death. Depends on the extent of damage. That is why it differs. If minor damage, DNA repair mechanisms will take care. If it goes beyond a certain step, cell growth arrest may be there. If it goes beyond that, it may go to apoptosis. So, like that, stage wise, depending on the extent of damage, things can happen. That is why you cannot just like that predict based on some agent. It depends on the strength of the agent, depends on the duration, it depends on the type of damage also done. So, there are many factors coming in. Of course, failure to repair double strand breaks result, we have said that they are repaired through, in this case, through the alignment of homologous sequences of DNA, because the homologous chromosome is there. So, that will be done, and when this happens during the S phase to M phase usually of the cell cycle, there are certain molecules which are required. That is where the difficulty comes in. I told you, whenever there is a damage, there must be some proteins to recognize the damage. So, that is one thing, and then for repairing you need some enzymatic activity, again you need some proteins, and after that of course the DNA polymerase and ligase. So, this will be the usual sequence of reactions. So, under that process you need some exonuclease activity, 3' 5' exonuclease activity, that is given by RAD50 MRE11 NBS1 complex, it is called. The complex has several components. So, that interestingly this also requires BRCA1. So, remember the BRCA1 is a gene associated with the breast cancer, and all of you know that if you have inherited some mutations in this gene, you are at high risk to develop breast cancer. You all know the famous example, one celebrity, who is that, Yes, Angelina Jolie. Yeah, she removed her breast thinking that she will get breast cancer. Of course, scientific opinion is that you need not have done that totally, but definitely there is some risk hereditary. But then remember, this is only in 5% of the cases, 95% are not hereditary. So, when you have these mutations, you are likely to have this one risk. So, that is not the point here. The point here is that BRCA1 is a tumor suppressor. So, that you may have to. So, the mutations in tumor suppressor are common in cancer. So, that way you can remember that. Here additionally, BRCA1 also participates in DNA repair mechanism. That is what is important, because you see, now you start connecting. DNA repair process works actually, here in this case it looks like it is working in tumor suppression. So, it will look like that, but if you take DNA repair process as such, it is if the DNA process is DNA repair process is very good in a person, what will be the effect of the anti-cancer agent? It will develop actually resistance, right? If the DNA repair process is down, then it will be chemosensitive. Now think about BRCA1. If it is normal, it will give you resistance, but if it is mutated, it is going to give you the treatment is going to be easier actually, because it is mutated, and so that will help to damage, because DNA repair process is actually down if there is a mutation. So, these are a little bit, it will look like conflicting, but if you think about it, then you will understand it. And another thing also you must remember, in BRCA mutation patients, if you give PARP1 inhibitors, both will act together, that will act as synthetic lethal to the system. You remember I told you what is synthetic lethal. This is the basis for that. At that time I did not explain. Actually, I forwarded one research paper on that, you can look at it. PARP1 inhibitors, if you put, they will that single strand break will turn into double strand break. Then BRCA1 mutation will further help, and that will result in double strand break, and finally that is how, if you have both, then the cells will eventually die. So, you can look at that, you can basically look at that paper and study that, then you will understand the mechanism very well. That is why I have attached one PDF file. For those of you who want to read more about that, you will understand. Another, whenever I found in the class, there is another confusion, that is about 6-O-methyl guanine. That also I have kept I sent a PDF file, look at it, because that is a source for confusion. The confusion comes in when I discuss that, probably I will tell you, but I will also just mention that 6-O-methyl guanine transferase, it is called MGMT, it is called methyl guanine methyl transferase. The problem in understanding that is the alkylating agents, they will methylate the guanine, methylate or alkylate, okay. But that alkylated residue will be transferred out of that by these enzymes. These enzymes will act only on methylated guanosines, because they transfer the methyl group. So, they will act. If you understand that point, then you will understand how a mutation of that will help in sensitization, chemosensitization. If you do not understand that, then there will be a problem in understanding how how the alkylating agents will be acting better when there is a mutation of that enzyme. So, only when you understand that, but when you read that paper, it will be very clear. You read that, then it will be easier for you to understand. Otherwise, it will be difficult for you to follow what we are saying. Anyway, here we are talking about homologous recombination repair. The 3' advancing strand from the damaged chromosome then pairs, because we have the homologous chromosome. As I told you, you will never understand unless we show the figure here. So, we will just see how this homologous recombination repair mechanism is operating. Let us imagine, see here, you see there is a double strand DNA break. So, both the strands are broken. Now, what will happen? There will be an exonuclease cutting at this place. Now, this will produce, what is that? It is called the free end. What do you call that? No, in molecular biology, what do you call that? Sticky end. So, it will result in the formation of sticky ends. So, there to the two sides you are having sticky ends. Now, what do you want to find? You must find a complementary sequence. Where the complementary sequence will be there? In the other copy. That is what you call as homologous chromosome. Simple. You just look at this. These are the two strands. I mean, not two strands, sorry, one strand, I mean one DNA only, but it is having a double strand. So, both the strands are broken, right. Now, let us think that you have the undamaged sister chromatid here. See, this is the undamaged chromosome. Now, you have cut it and you got this sticky ends, that is the first step. First step, creation of sticky ends, correct. You have some lengthy open arm, right, both sides. Now, what you need to do is, there are two sticky ends, right, here. You have to find some complementary sequence for that, for the DNA synthesis to happen. That is provided by the undamaged sister chromatid. We are assuming that one of the alleles is damaged, but it is two-stranded, double-stranded DNA, but one of the alleles is damaged. So, other allele is there. So, you are getting the complementary sequence from that. That is it. The matter ends there. Then the strand extension and disengagement and pair up and fill in the gaps with the DNA polymerase and DNA ligase. If you understand that, that is easy. So, all you need to know is that when there is a DNA break, double strand DNA break, the exonucleases act upon it and create long sticky ends in both the strands, right. And you find the complementary strand by just unwinding the normal copy, the other copy in the homologous chromosome. You unwind that, because for DNA replication, one of the first steps is to unwind the DNA, otherwise it is in coiled form. So, you have to unwind. You do not unwind the whole DNA, you always do it in bits and pieces, that you know, you must have studied in DNA synthesis, origin of replication and how that, what do you call that, origin loop and so on. They call it in electron microscope, they find some, I am also forgetting the name, focus what? Origin, replication foci and so on. Focus is singular, foci is plural. Not many people use that way, that is there. We like to say HIV virus, but HIV itself means virus, no. So, people always do that, does not matter. So, okay. If you understand that, this is very easy. The only problem comes in, what are the molecules that are involved. Here the process requires BRCA2 and RAD51. Then you have HR polymerase and yeah. So, these are the molecules that are required, and apart from that I also told you the other ones, that is this complex RAD50 complex. So, these are the three molecular components that are required for this. Now, there is another one. So, opposite to this one, non-homologous end joining. So, this is another process. We saw how this homologous, just to understand, it is based on the homologous chromosome. So, you have the other copy which is normal, from that you take the complementary sequence to repair that. Here it is non-homologous end joining. This means immediately you can understand, once you do this patch up work, the other one can be perfect, because you had all the sequences, but here if it is non-homologous, then it cannot be fully restored. You only do a patchwork. Suppose you have something like this, it is getting cut off. What I am going to do is cut this a little bit, bring it to shape and then join it. That is it. That means I am losing some portion. I am assuming that they are not that important. Even with this another 60-70% that is remaining, I will be able to do my work. This 30% that I lose, that is I am compromising. Some 30% loss I am compromising. 70% I am okay. If I am cured 70%, I am okay. I do not want 100%. That is what is non-homologous enzyme. I am just giving you the principle behind that. Again, it requires several enzymes and all that. First thing that you have to know is what is the double strand break that can be repaired by non-homologous end joining. So, that is ionizing radiation, bleomycin, topoisomerase 2 poisons and anthracyclines. Sorry. The molecules that are required for this process, especially the Ku70 and Ku80. Actually, Ku70 is some protein which is even like it is one of the anti-apoptotic factors also. If you look at the list, you will find that. But anyway, here it is a different purpose. DNA dependent protein kinase, DNA PK, and actually this Ku70 is part of the DNA PK. The DNA PK has regulatory subunit, catalytic subunit. So, this comes under this Ku70, Ku80 and all that. It is a heterodimer. That heterodimer is part of the regulatory subunit. What is this kinase? It is a serine threonine kinase. So, it belongs to the PI3 kinase family. So, what it does is it exhibits the sequence independent affinity for double stranded terminus and on binding to DNA and activates the catalytic subunit. The regulatory, it is just telling the additional proteins are also needed such as the Artemis protein, DNA ligase 4 and so on. Then what else is there? Yeah, the NHEJ, yeah, those who are familiar with immunology, they will know, has a normal function in immune cells to generate the diversity, immune cell diversity at the immunoglobulin and T cell receptor gene loci. This method is used. It is not that it is restricted only to DNA repair, that also you just understand. Now let us look at it because the picture will give a better story as usual. So, double strand break as usual in the same way as I told you in homologous, that is exonuclease, resection of the single strands by exonuclease, that will create the sticky ends, same thing. The only difference is after creating the sticky ends, the partnering sequence, if it is taken from homologous chromosome, you call it as that homologous this one. If it is non-homologous, then you call it as NHEJ, that is all. Now, you see from the same sticky ends, you look for some complementary sequence because it is a long sequence. So, you cut it in such a way that you will be able to pair with that, because there will be all these four bases only, A, T, C, G only, So, that can be paired up. That is very simple, because you see you do not see any other chromosome. It is the same DNA strand that was damaged that joins together. That is what I said. If you have a DNA and you break it here, I am only joining the same thing again, only thing it is double stranded, that is all. So, I am cutting off this portion 30% and joining. So, that means naturally I am losing some portion. So, that the other rest of the things are same, strands are filled in, joined by ligation, double helix is reconstructed. See here, several base, the last sentence is very important. Are you able to read that? Several base pairs which were present in the original wild type sequence are now missing. Naturally, when you cut and join, naturally you would have lost something. It is as simple as that. Okay, we are coming to the last one. Of course, I just told you there are 6 pathways, there are more pathways, but these are the most important ones that you have to remember. There are always several pathways. This is called translation DNA synthesis. is another mechanism especially designed for dealing with thymine dimers. You divide this chapter like that, you just understand what are the mechanisms that deal with single strand break, what are the mechanisms that will deal with double strand break. You also study what causes single strand break, what causes double strand break. If you read like that, if you try to read these slides, then it will be very easy for you to remember. Now, what is this mechanism? It is for thymine dimers and any bulky chemical adducts. This is what I said, DNA replication fork. You are saying some foci and all. What I wanted, that answer was replication fork. Replication fork will look like the bubble. They call it as bubble also. In electron microscope, you see that replication bubble also, they say. That is the terminology they use. What it means is, DNA synthesis does not take place just in a straight line. It takes place in bits and pieces. In these replication forks, which may be numerous, each one is first unwound and then the DNA replication takes place. Similarly, in 10 places it takes place and then you are joining. It is not like it is done continuously. That is the point I was trying to say. So, what will happen if there is a replication fork and you have a problem here because of the break, then it will stop. The regular DNA polymerase may not work, DNA polymerase delta, it may not work. So, the system, the cells will look for alternate polymerases, alternative polymerases. One such polymerase which is active in this translation DNA synthesis is called pol eta, polymerase eta, which will replicate past certain DNA lesions with high fidelity. The problem with polymerases is that there are about 15 DNA polymerases, but detailed mechanisms we are not very clear about it. We have several polymerases, we do not know each one of them in a very clear way. Detailed mechanisms are not known. Of course, the usual one is that DNA polymerase delta or whatever. Here in this case, for translation DNA synthesis, the pol eta will be utilized. Here you can look at it like I tell always, the picture. So, you have a lesion here, let us say a thymine dimer, then the thymine dimer is removed. A variant polymerase, that is what I told you, pol eta, is recruited to bypass the lesion, and then the usual process, DNA polymerase, replication and ligation, that completes the process. So, it is the same way, but only thing is, here it is DNA adducts, thymine dimers and so on, and they look for another additional polymerase. You may wonder why, sir, you are saying when you have the other pathway, like we have told, there is always alternate mechanisms are available. This is useful under certain circumstances, such as this thymine dimers. Sometimes more than one pathway can also be utilized. Now, basically DNA repair pathways will antagonize the activity of the anti-cancer agents, because you can understand if you think it in a simple way. Anti-cancer agent, its job is to do DNA damage. DNA repair mechanisms is to rectify that. It is a war between them. Whoever wins, he wins, that is it. DNA repair mechanisms means then the anti-cancer agent will not work. If the DNA repair mechanism is very efficient, then anti-cancer agent may not work. That is what you call as resistance. But if you want to sensitize the cells for anti-cancer agent, you make the DNA repair mechanisms defective. That is what is happening in BRCA1 mutation. BRCA1 helps in DNA repair, but if it is mutated, it is not going to function well. That means DNA repair mechanism is going to be defective. Now, the anti-cancer agent will work better. So, this is how it is. Now, you can make use of this also when you know what is the status of a cell or person in terms of DNA repair efficiency. You just look at the person, do some tests and see whether he has effective DNA repair mechanisms. Of course, I am telling it in a roundabout way. For example, even if the person is not having BRCA1 mutation, that means she is okay. I am saying she because it is usually in the women. So, okay. So, then we know that she is okay as far as DNA repair mechanism, at least normal BRCA1 is there. So, it will be okay. Like that, you have to do some diagnostic test. So, in other words, it may help to predict a suitable therapy for the patient. You look at whether the DNA repair mechanism is normal or it is defective. That will give you an idea about the suitability of the patient for treatment. So, that is one thing. Then what is that BER, NER short forms? That base repair and nucleic excision repair, NER pathways. Remove the DNA bases modified by alkylating agents. This MGMT, just now I told you, 6 methyl guanine DNA methyltransferase gene promoter methylation happens in gliomas. If promoter is methylated, what will happen to this gene? Correct, there will be loss of expression. So, that means this methyltransferase will not be expressed in brain cancer. Now, you take one patient in which this is expressed, one patient in which this is not expressed. Who can be better treated with alkylating agent? One in which it is absent. Okay. That is because this acts as an opposition to alkylating agents. Alkylating agents also methylate. This methyltransferase also transfers the methyl group. So, alkylating agents methylate and this fellow comes and removes the methylation. So, this MGMT removes the methyl group. That kind of facilitates the alkylating agents to methylate or alkylate. So, that is how they are doing the damage. So, that way that helps. Gene promoter methylation here, it silences the gene. That is what is seen in gliomas and colorectal tumors. Essentially switching off this enzyme, and this leads to the hypersensitivity, that is the word, hypersensitivity of these tumors to alkylating agents. So, this is a very important mechanism. That is why I gave you one paper to read. Just read that, you will clearly understand, because a beautiful figure is given how that removal and all that. In addition, damaged bases in the DNA can be bypassed through the use of TLS. TLS also can help. Rapid TLS is essential for a cell to go through S phase rapidly without replication, arrest and apoptosis. So, these are some of the mechanisms. TLS is also not a perfect process, it is an error prone process. So, use of TLS by cancer cells may result in mutation frequency also, that is also possible. Some of the I do not know what is this one, I also forgot. 15 variant polymerases can extend a nascent DNA strand. Okay. What he says is there are several polymerases that exist that can do this job. Pol-eta, this is the more important one. Pol-eta is also sometimes mutated in a particular disease called xeroderma pigmentosum. It is happening in the skin, where they found in some patients this Pol-eta is mutated. What it means is that this pathway is important, and if Pol-eta itself is mutated, then you get into some types of cancer. In this case, it is called xeroderma pigmentosum, reference to the skin actually. Absence of the Pol-eta enzyme results in UV light hypersensitivity. You know, these people, I think I mentioned that foreigners are very sensitive to UV radiation. Did I tell you? I have actually seen that. I worked in Israel and I don't know, maybe I mentioned it, I forgot perhaps. So, I saw that practically when we were doing these experiments with the UV rays. I was exposed for some time and nothing happened, but for the same time my friend, another Israeli, who is obviously white, he was suffering like anything within a few minutes. So, they are hypersensitive. Not that we won't suffer, long exposure will also have problem, but this what, so they are hyper sensitive. The absence of pol-eta enzyme is one problem. It is not that that particular person was suffering from pol-eta, this one, there may be lot of other reasons, but I am just saying hypersensitivity, inability to replicate past, this Thymic dimer problems, UV rays cause the Thymic dimers, that problem cannot be rectified if you have this pol-eta mutation. Ionizing radiation causes double strand breaks and oxidative DNA damage. Then HR repair, that is homologous recombination, NHEJ repair can deal with double strand break damage in cancer cells. So, these are all connected with that. So, basically tumors that have defects in these DNA repair processes are particularly sensitive to the cytotoxic activity of the ionizing radiation, because incidentally ionizing radiation we are using it for treatment also, right. Radiation can be used for treatment of cancer as well as radiation can cause cancer. That is why it depends on the dose, time and the area to which it is exposed, all several parameters, several variables are there. So, in general tumors that have defects in the DNA repair process are particularly sensitive to radiation. So, thus such people may be sensitive to ionizing radiation. They practically also, if you see, if you go to the hospitals, they will give different dosages. I took a relative who was very old, so they reduced the dose of radiation given, whereas for other people normally, also time. For example, if they give for I do not know what is the time they give, because I am not a doctor. So, they give, let us say, assume, I may be wrong, 10 minutes for somebody, they will give for 20 minutes for somebody. So, it varies depending on the age and the ability to tolerate that this one. The doctors would know by practice. So, they change that. Tumor cells that grow in a more hypoxic environment may also be more resistant. We just saw how hypoxia can contribute to resistance. So, that is also there. Killing of ionizing radiation due to the decrease in oxidative damage generated in these cells. DNA repair processes involve several other proteins also. One such proteins are the so called sensory proteins, that is DNA binding proteins, enzymes that remove the damaged bases and enzymes that restore the normal sequence. This is basically what I have already told you. That is, you need three things: proteins that bind the DNA, and proteins or enzymes that will remove the damaged portion, exonuclease, all those activities, and then enzymes that restore the normal sequence. What are those enzymes? Polymerase and ligase. So, that is what is shown here. Regulatory enzymes such as helicases serve to load DNA repair complexes at the sites of DNA damage. Helicases are also important. Topoisomerases, they unwind the DNA. These are all things that you study in DNA synthesis. Loading to chromatin and disassembly. In BER, a sumoylating, this is a new information. Sumoylation, you know what is sumoylation. Sumoylation is also another post-translational modification. Sumoylating enzymes modifies one of the glycosylases, thereby enhancing the activity of glycosylases in removing the damaged bases. Especially in this nucleotide excision repair mechanism, an E3 ligase complex activates polyubiquitination of the so called XPC protein. I am sorry, it is just so many details are there. It does not mean that you have to remember everything, but if you are interested, you can go through some of these names. There are too many proteins are involved. I also do not remember many of them. That is because in this system, each process involves, each of these DNA repair process involves a whole bunch of proteins. So, that is the difficulty. One is this XPC modification, that is in the NER complex. In translation synthesis, E3 ligase, I am just trying to list some of the molecules, RAD18, monoubiquitination, okay. PCNA, all these molecules are involved. Similarly, PP2A, which is a phosphatase. PP2A is a phosphatase, removes phosphates from what is called ATM. It is ataxia telangiectasia mutant, I think. That M I am also forgetting. So, ATM, that is very important. From ATM substrates, later on I will also tell you what is ATM, and there is one more, this one. Then there is a de-ubiquitinating enzyme called USP1. What is more important is the regulatory mechanisms involve so many number of these proteins, but if you have defects in these regulatory mechanisms, then that will result in failure to activate this DNA repair pathway or inactivate the error prone DNA repair pathway. So, this is the net result. In either case, the consequence may be higher mutation frequency, and ultimately you are predisposed to cancer. So, that is the most important consequence. There are other mechanisms also, like DNA crosslinks, interstrand DNA crosslinks, because when there is a damage, there can be a covalent modification of both strands. So, that also is another one. I think you do not have to worry too much about this. If you want, you can read this portion. I will give you the slides, but I think we will kind of stop here. This portion is damage responses is another area. I will try to teach that in the next class.